Good morning, children. So, I hope you have gone through the last day's class. We have completed the fatty acid biosynthesis. Only the long chain fatty acid synthesis, of course. The chain elongation has not been discussed yet. But what is the process with which or how is a fatty acid being synthesized in the body? So usually all the fatty acids synthesized primarily in the cytoplasm is of course the C16 palmitic acid, and we have gone in detail how is it being synthesized. So the synthesis involves three steps in fact. That is, first it has to be transported. The raw material, that is the acetyl-CoA, has to be the starting substrate, has to be first taken out of the mitochondria into the cytoplasm, which is carried out by the malate shuttle, followed by the carboxylation of the acetyl-CoA to give rise to malonyl-CoA, because it is not possible for two acetyl-CoA molecules to undergo condensation under a normal condition. It's thermodynamically unfavorable reaction. So in order to make the reaction a favorable one of the acetyl-CoA molecule will be shifted to a higher level, which is known as the malonyl-CoA, and it is carried out by simply the carboxylation. That is, we are adding one more carbon atom to it in the form of carbon dioxide or carboxyl group, so that the next higher member of acetyl-CoA is malonyl-CoA. So one of the acetyl-CoA molecules will be undergoing carboxylation reaction, which is again an energy dependent reaction carried out by one of the most important multi-enzyme system, which is known as acetyl-CoA carboxylase, and again it requires biotin as a cofactor or coenzyme. So that biotin first borrows the carbon dioxide from the bicarbonate ion, converting itself into carboxybiotin, and is carried out by carboxylase enzyme. Biotin carboxylase converts biotin or free biotin to carboxybiotin. This carboxybiotin, what happens? It then donates this carbon dioxide towards the acetyl-CoA, so that acetyl-CoA is converted into malonyl-CoA, while carboxybiotin will be converted back to free biotin. And again, this is happening by carboxybiotin transferase enzyme. Clear? So two enzyme activities are residing on a same molecule, and which is ultimately named as acetyl-CoA carboxylase. Clear? So that acetyl-CoA is converted into malonyl-CoA. So this is the starting. Or these are the initial steps which has to be carried out prior to the synthetic pathway, right? So first is the translocation from the mitochondria to the cytosol, followed by the carboxylation reaction. Now the acetyl-CoA in the form of malonyl-CoA is ready for undergoing chain elongation, and it's happening in the cytoplasm, and it is a multi-enzyme, multi-step process, but overall the reaction is carried out by single protein. But this single protein molecule is a huge molecule. It is having 7 different enzyme activities, each of the enzyme which is acting in an autonomous manner. So we can name simply as E1 to E7, right? But the E7 comes only at the last stage, right? When there is a thiolytic cleavage has to be taken place. Until then, E7 do not come into action. Only from E1 to E6, the reaction will be continued in a cyclic manner. So if it's a 16 carbon fatty acid which is to be synthesized, 7 cycles of synthetic reactions happens, and each cycle ends up with the formation of a fatty acid which is having 2 carbon greater than that of the substrate molecule. For example, in the first round, we are having one molecule of acetyl-CoA and one molecule of malonyl-CoA. Even though acetyl-CoA is coming in the form of malonyl-CoA, the third carbon, that is a carbon dioxide, will not be involved in the elongation step. Once the malonyl-CoA enters into the synthetic pathway, the first step again happening is a decarboxylation. So that ultimately there are reactions, the condensation between two acetyl-CoA molecules. Clear? So the starting substance was a 2 carbon compound, right? 2 molecules of 2 carbon compounds, right? Acetyl-CoA condenses together, and at the end of the cycle, once a single cycle is ended up, it results in the formation of a 4 carbon fatty acid, that is a butyric acid. Clear? So again, the butyric acid next enters into the next round of the cycle, and again the butyric acid will be condensing with malonyl-CoA, so that again butyric acid C4 will be converted into C6 at the end of the cycle, right? So this is how the fatty acid biosynthesis is happening. And again, the whole reaction require a very huge molecule which is known as a fatty acid synthase. It's a multi-enzyme complex having 7 enzyme activities, namely E1 to E7, right? Even though 7 reactions are there or 7 enzymes are there, the 7th enzyme will be coming only at the final cycle, the last cycle. 16 carbon fatty acid anengil, palmitic acid anengil, it enters into 7 round of synthetic reaction, 7 cycles, so that 16 carbon molecule will be formed. So only at the end of the 7th cycle, the E7 will be coming into action and detaches the fatty acid from the protein molecule. Until then, the thioesterase enzyme do not come into action. So only the E1 to E6 are coming into reaction in each cycles. Again, the first two steps are the we call it as a priming reactions. That is, we have to fix two pair condense cheyavenda. But two pair are coming, two molecules, two acetyl CoA, two substrate molecules are coming together, and each molecules are brought about by two arms, which is known as the central thiol arm and the peripheral thiol arm. So the first thing to be happened prior to the synthetic reaction is the bringing up of the two substrate molecules together. That is, the central thiol group should be occupied with an acetyl CoA or in the form of malonyl CoA, and the peripheral thiol will be also occupied with another acetyl CoA molecule. So it has to be first taken place. So that is the first two reactions. That is, we are fixing the acetyl CoA to the peripheral thiol. Peripheral thiol means cis-SH group, right? Cis-SH. That is carried out by, that is, acetyl CoA. From the acetyl CoA molecule, only the acetyl group has to be taken for the reaction. So the acetyl CoA molecule releases only the acetyl group, and it is going and getting fixed onto the cis-SH. This is carried out by an enzyme known as acetyl transacylase that forms E1, right? That is one of the priming reaction. And again, similarly, the malonyl CoA also comes, or malonyl group also comes in the same way as that of the acetyl group came, right? So in the case of malonyl CoA, the malonyl CoA also removes the malonyl group from it, and this malonyl group goes and gets fixed onto the central thiol group or the pan-SH, right? So now the pan-SH is occupied with the malonyl group, and the cis-SH is occupied with the acetyl group. So malonyl group is again transferred by another enzyme which is known as E2 or malonyl transacylase. So these are the two reactions which is happening in the very first steps of the synthetic step. That is the third stage of the synthetic reaction. Adim, translocation from mitochondria to cytosol. Second is the decarboxylation reaction. That forms the second stage. That rending kainal third stage is initiated, which is carried out by fatty acid synthase complex. A third stage, the first two steps on it is a priming reaction, in which the raw materials or the substrate molecules are kept ready for initiating the synthesis, right? രണ്ട് പേരും വന്ന് കണ്ടൻസ് ചെയ്ത് റെഡി ആയിട്ടൊക്കെ ഇരിക്കണം. Clear? So for example, if you have a university exam, first you are supposed to collect the hall tickets. Hall ticket collect your marks has to be uploaded first. We are checking whether you are eligible or not for appearing the university exam. That is the first stage. After that, if you are crossing this stage successfully, if you are getting promoted for entering into the university exam, then you will be allotted with a hall ticket, right? So that is the second stage. Now, once you are having your hall ticket with you, now you are having the permission to enter into the examination hall, right? And once you enter into the examination hall, what happens? You are allocated with a position, right? So the registered number, the seat marked with your registered number is your location. So that is a priming reaction. You are getting seated on your corresponding seat, right? So that's a priming reaction. Only then you will be given the question paper or the reactions happening. Clear? So that's a priming reaction. What? You are getting allocated a seat for you. So you should be seated only on your seat, not another person's seat, right? In the same way, the acetyl CoA molecule is seated in only the cis-SH arm, while the malonyl group will be, it's not the acetyl CoA, it's only the acetyl group. Acetyl group is coming and getting fixed on, or it is getting seated on the cis-SH arm, while the malonyl group will be getting, or it is allocated with the central thiol group or the pan-SH group. So the pan-SH, it is getting fixed onto the pan-SH arm. But end arms are now occupied with our substrate molecule, acetyl-CoA in the peripheral arm and malonyl-CoA in the central arm, right? And again, they are coming together. They are seated adjacent to each other. Now they are ready for initiating the synthetic reaction. And the synthesis starts with what? First is a condensation reaction. Condensation is followed by the reduction reaction, reductase enzyme. Again, the reduction is carried out by NADPH. Then comes again the dehydration reaction, the removal of the water molecule or introduction of a double bond. And comes the last stage, again, the second step of the reduction reaction. Again, it is carried out by NADPH. So once this 4 steps are completed, what is happening? The 2 molecules are getting condensed. Clear? So condensed so that the 2 carbon molecule is now upgraded to a 4 carbon fatty acyl CoA. But this fatty acyl CoA is located on the pan-SH arm, right? So only the malonyl CoA always comes and gets fixed onto the pan-SH arm or the central thiol group. So in order to initiate the second round, what is to be done? This elongated or this bigger molecule now residing on the pan-SH has to be translocated from the central thiol group or from the pan-SH. It will be shifted towards the cis-SH. Clear? So that now this pan-SH is free and the next molecule of malonyl CoA or malonyl group can come and get fixed onto it and the reaction can be repeated, right? So this is how the fatty acid biosynthesis is taking place. I hope you have understood it. So this is a nutshell, or this is the stoichiometry of the reaction. That is 7 cycles are happening because we are having 16 carbon fatty acid with us. So 16 carbon means we require 8 molecules of acetyl-CoA. Out of the 8 molecules, only a single acetyl-CoA is coming in the form of acetyl-CoA SH. All the remaining acetyl-CoA molecules, remaining 7 molecules of acetyl-CoA molecules are coming in the form of malonyl-CoA, right? And all these malonyl-CoA will be coming and getting fixed only on the pan-SH or the central thiol group. Okay, so acetyl-CoA, 1 molecule of acetyl-CoA condenses with 7 molecules of malonyl-CoA in presence of 14 molecules of NADPH, or by consuming 14 molecules of NADPH and 14H plus gives rise to what? The C16 palmitic acid plus 14 NADP plus 7 carbon dioxide. That is, we are having 7 molecules of malonyl-CoA, and at the first step of the synthesis itself, that is during the first condensation reaction, what is happening? The carbon dioxide is removed, right? 7 carbon dioxide molecules with 8 CoA molecules, right? ഇവിടുത്തെ malonyl group റിലീസ് ചെയ്യും കോയനെ. അപ്പൊ 7 malonyl ഗ്രൂപ്പിന്റെ CoA-SH ഉം ഒരു acetyl CoA-യുടെ ഈ CoA-SH ഉം. So 8 CoA-SH, right? And 6 water molecules, right? So all these are the reactions happening during one round of fatty acid synthesis. One round for the synthesis of 1 molecule of 16 carbon fatty acid. But again, we know that a fatty acid synthase complex is it's a dimeric molecule which is constituted by 2 sets of monomers, right? So at the same time, simultaneously, we can have 2 molecules of palmitic acid, right? So this is how the palmitic acid is synthesized. And we can just compare between beta oxidation, catabolism and anabolism compare. We know that beta oxidation is a mitochondrial process, while fatty acid biosynthesis or the de novo pathway is cytoplasmic in nature. Then the carrier molecules, coenzyme A and acetyl-CoA, acyl-CoA, it's a carrier molecule, while here we are having the cis-SH and the pan-SH. Right, here the electron acceptors or the reducing equivalents are FAD and NAD, while here we are having 2 molecules of NADPH. Then hydroxy intermediates, the water molecules are coming in the form of L form. Here it is in the D form. Then the product obtained is oxidation we are having acetyl-CoA. Synthesis we are the acetyl-CoA condense only out of the 8 molecules, only a single molecule is coming in the form of acetyl-CoA. The remaining molecules are coming in the form of malonyl-CoA. Clear? So this is a comparison between the 2 processes. This is the reverse. Okay, right. Now, next we are going to discuss how is the fatty acid biosynthesis regulated. See, ഹൗ ഇസ് ഇറ്റ്, കാരണം അത്രയും കോംപ്ലിക്കേറ്റഡ് ആയിട്ടുള്ള എൻസൈം മോളിക്യൂൾ ആണ് ഫാറ്റി ആസിഡ് സിന്തേസ് ആണെങ്കിലും അസറ്റൈൽ കോയെ കാർബോക്സിലേസ് ആണെങ്കിലും. സോ ഇറ്റ് ഈസ് അണ്ടർ സ്ട്രിഞ്ചന്റ് റെഗുലേഷൻ. ഫാറ്റി ആസിഡ് എക്സസ് ആയിട്ടുള്ള കാർബോഹൈഡ്രേറ്റ്സ് വന്നാലും എക്സസ് ആയിട്ടുള്ള പ്രോട്ടീൻസ് വന്നാലും എക്സസ് ആയിട്ടുള്ള ഫാറ്റ് വന്നാലും ഇതൊക്കെ എന്താ ചെയ്യാം, എല്ലാ എക്സസ് ആയിട്ടുള്ള നിന്നും ബോഡി ഒന്നും കളയില്ല, ഒന്നും വേസ്റ്റ് ആക്കില്ല. എവറിത്തിങ് വിൽ ബി സ്റ്റോർഡ് ഇൻ ദ ഫോം ഓഫ് ട്രൈഅസൈഡ് ഗ്ലിസറോൾ. അപ്പൊ ടി ജി ഉണ്ടാവണമെങ്കിൽ എന്ത് വേണം, ഫാറ്റി ആസിഡ് വേണം. അപ്പൊ ഫാറ്റി ആസിഡ് ഹാസ് ടു ബി സിന്തസൈസ്ഡ്. സോ ഹൗ ഇസ് ദ ഫാറ്റി ആസിഡ് സിന്തസിസ് regulated?